Alright, let's learn about mitosis. To do that, I'm going to need your undivided attention. If you didn't get that joke, then you are in the right place, because you obviously know nothing about mitosis. There's a lot we know about the universe and a lot we don't. In 1872, a scientist named Walter Fleming used aniline dyes to stain the cells of a salamander embryos. The dyes gave him the ability to visualize the division of cells. The discovery is considered one of the 100 most significant scientific developments of all time and the top 10 of the most important discoveries in the field of biology. This became known as mitosis. Mitosis consists of 5 phases. Although some don't consider interphase to be part of mitosis due to it being the normal life part of the cell lifespan. In this case, we're going to include it because it's an important step in mitosis. The scientific definition of mitosis is when one cell divides to form 2 identical cells. For this process to begin, we must start with the prologue of mitosis, called interphase. This is the longest phase. During this phase, the cell prepares for mitosis. This includes DNA replication and growing weight or mass. Within the phase, there are 3 phases. Wow, like the DOS Day effect, right? But for science. The 3 phases include the G1 phase, the S phase, and the G2 phase. The G1 phase happens prior to the replication of the DNA. During this time, the cell increases its mass to prepare for the upcoming division, like a bear getting ready for hibernation. The G1 phase represents for the gap1 phase. Scientists are so creative, right? The second phase in interphase is the S phase. This is when the DNA is synthesized. This phase is the smallest of phases, happening in a small time window. And the S stands for synthesis. The third and final phase of interphase is the G2 phase or gap2 phase. This happens after DNA synthesis. This late into interphase, the cell still has nucleoli present. The chromosomes are duplicated but are in the form of chromatin. The nucleus is bounded by a nuclear envelope. No, not that kind of, this kind. Trust me, this is meant to be like a prison, keeping the sister chromatids in. Now we start with the prophase. During this stage, the nucleolus fades and the replicated DNA or chromatin condenses into chromosomes. The mitotic spindles will form in the cytoplasm. Get out your letter opener because this late into the prophase, the nuclear envelope breaks up, releasing the hounds, which starts to migrate towards the center of the cell. Thus begins metaphase, in which the nuclear membrane also disappears. During metaphase, the sister chromatones will align in a straight line in the center of the cell, preparing for anaphase, in which they start pulling apart the sister chromatones toward the 2 different poles. The sister chromatones break apart, each pair being the exact copy of each other. The nucleoli begin to form at the opposite poles. The 2 new parts break apart to create 2 new cells known as the daughter cells, which both have exactly the same number of chromosomes as the parent or original cell. And that was the beautiful cycle of mitosis. I hope you enjoyed this educational video that you'll probably leave wondering how a girl can make so many horrible jokes on a subject like mitosis. Well, I have one thing to say in response. What did one cell say to his sister cell when she stepped on his toes? Mitosis! And I forgot. If you didn't get that joke, then you weren't paying attention at all to this video, because, come on, that was pretty great. Alright, I get it. Thanks for watching, and I hope you enjoyed this lesson on biology, the only place where division and multiplication mean the exact same thing.